Hi students, today I am going to continue the topic that Mendelisms. In the previous session, I have discussed about the introduction to the genetics. Okay, the previous session நம்ம ஒரு சில terms வந்து understand பண்ணியிருப்போம். What is gene? The gene என்ன? It is an inherited factor. So இது மரபணுன்னு சொல்லுவாங்க. இந்த மரபணு தான் ஒரு பேரண்ட்ஸ்லேருந்து ஆஃப்ஸ்பிரிங் போகிறது. மூதாதையர்களேருந்து இளம் சந்ததியினருக்கு வந்து அந்த மரபணு டிரான்ஸ்ஃபர் ஆகுது. இதை பற்றி நிறைய டிஸ்கஷன் பண்ணியிருப்பாங்க மெண்டலியன் தியரியில். இந்த மெண்டலியன் தியரியில் வந்து நிறைய கான்செப்ட் வந்து அந்த ஃபிலாசபர் சொல்கிறத மக்கள் பிலீவ் பண்ணாங்க. பட் இட் இஸ் நாட் அ கிளியர் ஒன். இந்த 18th 19th செஞ்சுரி வரைக்கும் இந்த வாட் இஸ் ஜெனetics அப்படின்னாலும் என்னன்னு தெரியாது. ஸோ மோஸ்ட் ஆஃப் த சயின்டிஸ்ட் தே ஆர் thinking about how it is able to transfer the characters from the parents to offspring. ஸோ எப்படி அந்த கேரக்டர்ஸ் வந்து பேரண்ட்ஸ்கிட்டேருந்து ஆஃப்ஸ்ப்ரிங்க்கு போகுது, எப்படி ஒரே நேச்சர் ஒரே ஐடென்டிக்கலாக இருக்குது அந்த பிளான்ஸ், அந்த பேரண்ட் பிளான்ட்டும் ஆஃப்ஸ்ப்ரிங் பிளான்ட்டும், இல்லைனா பேரண்ட்டும் அந்த ஆஃப்ஸ்ப்ரிங் பேட்டர்னல் அண்ட் மெட்டர்னல்ஸ் இந்த ரெண்டுமே எப்படி வந்து சேமாக இருக்குது, அந்த ஆஃப்ஸ்ப்ரிங் எப்படி சேமாக இருக்குது. ஸோ இது எது வந்து இந்த கேரக்டர்ஸ் வந்து ட்ரான்ஸ்ஃபர் பண்ணு. டுடே we know that the gene is responsible for the transmission of the characters from parent to offspring. So in 18th and 19th century they were not clear about which is causing the transmission of the characters. சரிங்களா? ஸோ அந்த மெண்டலியன் தியரியில் நம்ம அரிஸ்டாட்டில் சொன்னதை பார்த்துருக்கோம். ஓகே, அரிஸ்டாட்டில் வாட் he proposed the theory called that is a reproductive blood theory. And the reproductive blood theory la enna solrena that the pure reproductive blood of the male, that is semen, fuses with the impure reproductive blood of female, the menstrual flow, to form offspring. That is the concept. But this Mendelian theory was not giving the scientific idea. Okay, so then when the Mendelian period starts, so Mendelian is Gregor Johann Mendel is one of the eminent scientists actually. So during his lifetime he was not recognized by the people. அவருடைய கான்செப்டை யாருமே ஏற்றுக்கல. He was telling the factor is responsible for the transmission of characters from the parents to offspring. Okay, until after his death only 3 scientists, the eminent scientists, they were rediscovered the Mendelian theory and proved the Mendelian theory was perfectly correct. It has given it opened the window for the transformation of the genetics field. Okay, so Mendel, Gregor Johann Mendel, he was an Austrian monk. Okay, we know that he's a Christian priest, Austrian monks. When we study about his lifestyles, his life, okay, he was born to the very middle class family where he was not able to afford much money for the education, but his father wanted him to be a teacher. Okay, he also tried and written exam, but he could not pass in that teacher examination. Okay, so finally he decided to join in the monastery, Augustine monastery in Brunn. Okay, after joining the monastery, so அந்த monastery join பண்ணி still his thirst for the science and maths is not dissolved. அவருடைய அந்த thirst அதில் வந்து சாதிக்கணுன்ற ஒரு வெறி இருந்ததால் he was continuously learning. That's why we call he is a lifelong learner. So when we describe about the Mendel, Gregor Johann Mendel, we how to call him is a lifelong learner. He's a scientist, he's a monk. Okay, he is an actually natural scientist. So he was having a lot of interest in the science and mathematics. So why his theory became famous and why it became successful? Because he introduced the mathematical knowledge in his scientific temper, in his scientific research. So he combined mathematics and science and given the accurate data regarding many progenies experiments. Okay, so that's why his research, his theory is accepted by the world and it has given the new window to do more research in the genetic field. Okay, so this Austrian monk, he was born in 1822. Okay, Then later on he studied well enough and he joined in the Augustine monastery. Monastery, what is monastery? Monastery is ஒரு ஆன்மீகவாதிகள் தங்கி படிக்கிற இடம். Only they learn about the religious activities. That's we call it an monastery. So there, though he was doing the priesthood and he was showing much interested in finding out what causes the transmission of the character from parent to offspring. Why he got that much interest in that? Because during his period, there was a prevalent theory discussed in the society, that is an inheritance of blending. நம்ம முன்னாடி கிளாஸ்ல பார்த்துருப்போம் inheritance of blending. What is inheritance of blending? The characters of parents got fused or mixed up and it passed to the offspring. The characters of parents are got mixed up and it passed to the offspring. So that was the inheritance of blending theory. So during the Mendelian periods, there was a wide accepted theory that's called inheritance of blending. Inheritance of blending, the characters of the parents got fused or mixed up or blended to form an offspring. So in the parents ude character, male female parents ude character mixed up ஆயிருக்கு. So அதுதான் அந்த ஆஃப்ஸ்பிரிங்ல வந்து எக்ஸ்பிரஸ் ஆகுதுன்ட்டு அந்த inheritance of blending theory explains to us. Okay, so that he got that Mendel, Gregor Jürgen Mendel got curiosity. Okay, when he observed in his garden pea plants, he was not able to prove that this Mendelian, that inheritance of blending is correct. So he was finding out that was not correct, because the tall plant always gives the tall offspring. The short plants always give that short offspring. There was no middle intermediate height identified in the garden pea plants. So then he got an intention. So he thought that then what will be the reason? What can be the reason? What can be the another concept which transmit the heredity, which transmit the characters from the parent to offspring? So thereby, when he was doing experiments in garden pea plants and he was able to propose the rules of inheritance in 1865. So in 1865, he has proposed the rules of inheritance based on the experiments conducted in the pea plants with the help of the techniques called hybridization. Okay, so he has conducted experiments in the garden pea plants, that is pisum, sativum. Okay, and based on that hybridization method. So in 1865 he has come forward with the rules of the inheritance, but in 1866 he described in his paper called that is an experiments on the plant hybridization. So in his paper he explained how this inheritance, the law of inheritance will be, how this transmission of characters take place. So அவருடைய புக் வந்து experiments on the plant hybridization என்ற புக்ல வந்து அவர் தெளிவா அவருடைய rules of inheritance பத்தி explain பண்ணிருக்கு. Okay, so it is available in the 4th volume of annual proceedings of natural history and natural society of Brune. So அந்த ஜர்னல்ஸில் தான் இவருடைய பேப்பர் வந்து பப்ளிஷ் ஆனது. But most of the scientists, they did not accept his concept. He is blabbering and that will not be the real concept. So during his lifetime, he was not recognized for his work, his wonderful work. Okay, none of them recognized his splendid work. So thereby in 1884 he died. So after his death, there are 3 eminent scientists has taken back his laws and identified the Mendelian law is perfect and it is the basic for the genetics. So who are the 3 scientists? So one scientist name is Hugo de Vries. Okay, Hugo de Vries and Tishmar and Carl Corrins. These are the 3 scientists. They have done the experiments on different model system and they have concluded the Mendelian theory was perfectly correct. The factors are responsible for the transmission of the characters. So everyone accepted in 1900s. So So these three scientists who are all given the proof to the Mendelian theory, they are given the laws called Mendelism. Now that Mendelian theory is known as Mendelism. This law is called as a Mendelism. Okay, then what are the experiments that Gregor Johann Mendel conducted? How he concluded, how he has proposed his inheritance theory? Let's see one by one. Gregor Johann Mendel selected his model system. So he has selected the plant, the model system, pisum, sativum. Okay, this is garden pea plant. Okay. This is the model system of Mendel experiments. Okay, so Mendel has selected a simple plant, that is a garden pea plants. That's the scientific name is pisum sativum. He has selected his model system for these experiments. In the previously he selected honeybee for his experiments. Okay, so which was not worked out properly, and finally he come to the small plant, the garden pea plant. Because why he has selected this garden pea plant? Yeah, in the garden pea plant அவருக்கு ஒரு சக்சஸ்ஃபுல்லான எக்ஸ்பெரிமெண்ட் கொடுத்துச்சு. Because these garden pea plants have a 7 contrasting traits. 7 விதமான characters visible இருக்கு. Okay, 7 contrasting traits are visible. So phenotypically visible. So these 7 contrasting traits help that Mendel to study more and more, and help the Mendel to conduct his experiments very faster. Okay, so 7 contrasting trait. So what will be the 7 contrasting trait? So height of the plant, flower color, the position of the flower, okay, the seed ன்னு பார்க்கும்போது அதனுடைய ஷேப், அதனுடைய சைஸ். Okay, play. So height of the plants, flower color, position of the flower, the seed, shape and size. So along with it, the pod, color and the size. Got it? The height of the plant, the flower color, the position of the flower, the seed, shape and size. Okay, and we can have the color also, color of the seed. Then the pod is the outer covering. Inside that we will identify the seeds. Okay, so bean seeds, if you take bean seeds, then outer covering are the pod and one. அதுக்குள்ள இருக்கிற அந்த seeds. Okay, the seeds are connected very closely in the pods, and the pods closes and protect the seed. So அந்த pod color பார்த்தீங்கன்னா டிஃப்ரெண்டாக இருக்கும். green color இருக்கும். Okay, then அது flattened இருக்கும், bulged outல இருக்கும். All these things. Got it? So these are the 7 contrasting traits are visible in the garden pea plants. So அவர் அந்த எதனால் அந்த எக்ஸ்பிளைன் அந்த கார்டன் பீ பிளான்ட்டை அவருடைய மாடல் சிஸ்டமாக எடுத்தது காரணம் இதான். There are 7 different contrasting traits are visible in the pea plants. Got it? The next one. The lifespan. The lifespan of the cotton pea plants is very short. Okay, so 3 months. So 3 months plants. So he can harvest a huge quantity of seeds. He can do his experiments as much as time possible. So nariya experiments pannalam. Plus he can get a huge quantity of the seeds for his experiments. Okay, so that is another reason why he is selected. The third one, the chromosome. Some of pea plants is 14 in diploid condition. Okay. So next thing is, so we have discussed about the first thing is 7 contrasting traits. Next one, the lifespan. Okay. Then the chromosome of pea plants. The chromosome of pea plants. Then next one is a genetic makeup. Huge quantity. Okay. Then quantity of seeds can be collected. The foremost very important thing is self-pollination. Self-pollination. Okay, autogamy. We call it an autogamy. Do you understand why he has selected this garden pea plants? First thing is it shows 7 different contrasting. 7 contrasting traits. 7 genes are identified for 7 contrasting traits. Okay, the lifespan of the pea plants is very short, 3 months plants. So lifespan of the pea plants will be shorter. So 3 months plants. So thereby he can collect, he can harvest so many seed as much as possible. அது மட்டும் இல்லாம நிறைய எக்ஸ்பெரிமெண்ட் கண்டக்ட் பண்ணலாம். Okay, he can conduct many experiments. So around roughly he has conducted experiments in 30,000 pea plants. 30,000 pea plants. So 30,000 pea plants la அவருடைய எக்ஸ்பெரிமெண்ட்ஸ் அவரை கண்டக்ட் பண்ணி அதனுடைய ரிசல்ட்டை வந்து கால்குலேட் பண்ணிக்கிறார். Okay, then the lifespan of the pea plants is very shortest and he can cultivate as much as time possible. Okay, he can collect large quantity of seed for his experiments. Okay, and next the chromosome pea plants is in 14 in diploid condition. The genetic makeup is also helping out and giving the shortest way of finding out the transmission of the character from the parents to offspring. And self-pollination, autogamy. So self-pollination is one of the advantages. Okay, so which will give the pure breed, pure line of the parents. Got it? Clear? So these are the things, are the characters which made the Gregor Johann Mendel to select the cotton pea plant. Okay, so his Mendelian experiments has 4 steps. Okay, Mendelian experiments. 4 steps. Number 1, selection process. Okay. Number 2 is an hybridization. Number 3 is selfing. Okay. Number 4 is calculating. These are the 4 stages are followed in the Mendelian experiments. Selection. He selected the garden pea plants and he has gone for the hybridization process. Then allowed this F1 progeny, self-pollination, okay, or self-fertilization process. Then finally he collected the seed and these seeds are calculated for the variations. Okay, these are the 4 steps followed in the Mendelian experiments. Let's move on to the Mendelian experiments. Okay, so Mendelian experiments is Mendel, Gregor Mendel. He has connected his experiments with limited traits every time. Okay, he connected his experiments with limited traits. He did not get confused by using more traits at a time. So little by little he improved his experiments. Okay, so what is it? So he has taken the pure breed. Okay, so pure breed example, if you take the tallness, he has taken the tall plants and crossed with the dark plants. Pure breed, dwarf. Okay, dwarf. So already we know that each characters, the trait having the two factors are the two pair of allele. Okay, T and T. These are the factors. Okay, and this is expressed small t. These are the factors for the tallness. Okay, these factors determine the character's tallness. So this is a pure breed. So when he has taken the pure breed and he allowed for the self-pollination process. Okay, or pure breed or parent, pure parent plant pea plant அது வந்து self pollination allow பண்ணார். So he has studied many generations. First parent எடுத்து அந்த pure இப்போ tall plant எடுத்தேன்னா அந்த tall plant self fertilization self pollination allow பண்ணி அதுக்கப்புறம் அதனுடைய progeny எடுத்து திரும்பியும் cultivate பண்ணாரு. அதே தான் வருது. Okay, a pure parent, so it is able, we can easily able to find out the pure breed. So pure breed எடுத்து அது எத்தனை தடவை நாம வந்து self pollination எத்தனை generation பார்த்தாலும் அந்த pure breed what the parent is having that character only shown out. அதே போல தான் இங்க dwarf characters எடுத்து அந்த characters இருக்கிற அந்த plant எடுத்து pea plant எடுத்து அது வந்து self pollination allow பண்ணனும். How many generation it goes on and it will have only dwarf characters. So that shows it is an 100% pure breed. So in the pure breed இப்படியே போனா அவருடைய experiments so வந்து கண்டுபிடிக்க முடியாது. So what he has done, he wanted to do hybrid. The two different traits வந்து cross பண்ணனும். Two different traits cross பண்ணுது. So he has taken one pair of contrasting trait. So characters இதுல trait என்றது என்னது is a character. Okay, trait என்றது characters. So he has taken one pair of contrasting traits. So contrast. So ஒரு parent வந்து tallness ஆ இருக்கு, இன்னொரு parent வந்து dwarfness ஆ இருக்கு. So is the height of the plant. This is the height of the plant. So he has taken the character height. Okay, he has taken the character height. Got it? So height எடுத்துக்கிறேன். Height எடுத்துட்டு அதுல வந்து ஒரு parent வந்து ஒரு pea plant ஒரு டாலா இருக்கு, இன்னொரு pea plant வந்து டார்ஃபா இருக்கு. So இந்த ரெண்டையும் எப்படி cross பண்ணனும்? So he has brought one artificial method of cross pollination. Artificial pollination பண்ணு decide பண்ணிட்டார். Okay, so he has gone for the procedure called emasculation. The emasculation என்ன? What is an emasculation? இப்போ ஒரு pea plantல, okay, so this is one pea plant and it will have the both male and female gametes in the flower. That's why it is going for the self pollination. So pea plants and the garden pea plant எடுத்துட்டு என்ன பண்ணு ஒரு ஃப்ளவர்ல இருக்கிற அந்த ஆந்தரை வந்து ரிமூவ் பண்ணிடுறாரு. ஓகே, ஸோ இப்போ வந்து he has taken the tall plants, tall pea plant எடுக்கிறாரு எக்ஸாம்பிள், அதில் இருக்கிற அந்த ஒரு ஆந்தரை வந்து ரிமூவ் பண்ணிட்டு ஸ்டிக்மா வந்து வைக்கிறாரு. ஓகே, அட் த பட்டிங் ஸ்டேஜ். அதே போல் இன்னொரு பிளான்ட் எக்ஸாம்பிள் டார்க் பிளான்ட் இருக்கு இல்லையா, இதில் இருக்கிற அந்த வந்து ஸ்டிக்மாவை எடுத்துட்டு ஆந்தரை மட்டும் வைக்கிறாரு. ஸோ வாட் ஹேப்பன்ஸ் நவ், he has taken two different flowers of the same species. Okay, so ஒரு ஃப்ளவர்ஸில் வந்து ஆந்தரை ரிமூவ் பண்ணிடுறாரு, இன்னொரு ஃப்ளவர்ஸில் வந்து ஸ்டிக்மாவை ரிமூவ் பண்ணிடுறாரு. ஸோ இப்படி ரிமூவ் பண்ணிட்ட பிறகு இது கவர் திஸ் பட். ஓகே, அந்த ரிமூவ் பண்ணிட்ட பிறகு அந்த ரெண்டு ஃப்ளவர்ஸையும் பாலித்தீன் பேக் வச்சு கவர் பண்ணிடுறாரு. Why he is covering? He is Why he is covering? There may be another pollination or cross pollination, or that pollen grains from the another species may drop there. Okay, if it is dropped, then he can't find out. அவரால் அந்த எக்ஸ்பெரிமெண்ட்ஸை வந்து கம்ப்ளீட் பண்ண முடியும். He will not get a positive result. அதனால் once before doing the hybridization process, அந்த ஹைபிரிட் ப்ராசஸ் போகிறது முன்னாடி he was doing emasculation process. இந்த emasculation ப்ராசஸில் ஒரு ஃப்ளாரில் இருக்கிற அந்த ஆன்தரை ரிமூவ் பண்ணிவிட்டு அந்த ஸ்டிக்மாவை மட்டும் கவர் பண்ணிடு. ஓகே, அந்த ஃப்ளாரை பாலித்தீன் பேக்கை வச்சு கவர் பண்ணிடு. அடுத்த இன்னொரு ஃப்ளாரில் அதே போல் அந்த ஸ்டிக்மாவை எடுத்துகிட்டு ஆன்தரை மட்டும் வச்சு அந்த ஃப்ளவர் கவர் பண்ணிக்கிறேன். allow for the blooming of the flowers. அந்த மெச்சுரேஷன் நடக்கணும் இல்லையா? ஓகே, இந்த ஓவியூல் மெச்சூர் ஓவியூல் வந்து கொஞ்சம் பெருசாகணும். அதே போல் அதில் போலன் கிரெயின்ஸ் சைஸ் கொஞ்சம் பெருசாகணும். ஸோ he allows for the certain periods. Once that flower, the bud is bloomed into flower, what happens? He takes the pollen grains. Okay, ஸோ ஒரு செடியில் ஒரு பிளான்ட்டில் இருக்கிற pollen grains எடுத்து அதே பிளான்டோடைய ஃப்ளார்ஸில் ட்ரான்ஸ்ஃபர் பண்ணிடு. Okay, from one flower he has taken. So he has taken the pollen grains and he transferred to the stigma of the flower. Okay, after transfer பண்ணிட்டு வரக்கு what happens? This is called cross pollination. ஏன்னா cross pollination நேச்சுரலாக நடக்க வாய்ப்பு கிடையாது இந்த பீ பிளான்ட்டில், ஆர்டிஃபிஷியலாக தான் நடக்க முடியும். ஸோ அதனால தான் அவர் அந்த எமாஸ்குலேஷன் ப்ராசஸ் மூலயமா அந்த போலன் கிரெயின்ஸை ஸ்டிக்மாவுக்கு ட்ரான்ஸ்ஃபர் பண்ணிட்டார். Now, a pair of contrasting traits are involved in the fertilization process. So, pollination is a primary stage of the fertilization. So, fertilization now that flower developed into fruits and it is having the seed. And the seed எடுத்து அவர் திரும்பி கல்டிவேட் பண்ணி பார்க்குறாரு. When he cultivated, to his surprise, he got only one trait. He has taken a pair of contrasting trait. Okay, ரெண்டு விதமான ட்ரேட் எடுத்துக்கிறாரு. a pair of the tallness, darkness, இந்த ரெண்டுத்தையும் எடுத்து கிராஸ் பண்ணி ஹைப்ரிடைசேஷன் பண்ணி அந்த ஹைப்ரிட் அந்த ப்ராஜெனியை வந்து கல்டிவேட் பண்ணி பார்க்கும்போது அந்த ப்ராஜெனியும் ஏதோ ஒரு ட்ரெயிட்டை தான் ரெப்ரசன்ட் பண்ணுது. ஓகே, ஸோ தட்ஸ் அந்த ரெப்ரசன்டட் ட்ரெயிட், அந்த எக்ஸ்பிரஸ் ட்ரெயிட் இந்த F1 progeny according to him he called it dominant. So one trait is masking over of the another trait. ஒரு trait வந்து இன்னொரு trait, ஒரு ஃபேக்டர் வந்து இன்னொரு ஃபேக்டர் வந்து கவர் பண்ணுது. ஸோ the factor which mask over the another factors and expressed phenotypically. இங்கே phenotype genotype என்னன்னு தெரியணும். வாட் இஸ் phenotype and genotype? நான் 7 contrasting trait ன்னு சொல்லணும் பார்த்தீங்களா, அந்த கலர், the physical appearance of the plants, physical appearance of the plants நம்ம phenotype ன்னு சொல்லுவோம். Physical appearance of the plants we call it in phenotype. Okay, and the genetic makeup நமக்கு தெரியாது. Okay, so the genetic makeup of the plants we call it in genotype. So you should know the term phenotype and genotype. External appearance of the plant is known as a phenotype characters. Okay, internal makeup of the gene is known as genotype. Okay, அவரு அந்த டால்னஸும் டார்க்னஸும் இந்த ரெண்டு பியூர் பிரீடை எடுத்து கிராஸ் பண்ணும்போது நீங்க F1 progeny-ல is able to identify only one of the trait. So this is a tallness and this is a darkness. So the factors for a tallness. So he has taken one pair of contrasting trait, one pair of contrasting trait for hybridization. So this is, we call it monohybrid. Only one pair of traits is taken for the experiments. Okay, so what is monohybrid? Two different parents, two different pure parents are crossed. Okay, or else you can take it one pair of contrasting traits of for hybridization is known as a monohybrid. So the pure breed irukku, tallness, is pure breed dwarfness irukku. So we have taken only a pair of contrasting, only one pair of contrasting trait. So tallness contrast is dwarf. So we have taken only one pair of contrasting traits for hybridization. So this is known as monohybrids. So when we have taken and done the emasculation, cross pollination. So cross pollination allow pantaru. Okay. So when he is doing the cross pollination, then he is able to get okay. This is a parent representation P, that is a parent pure breed. Okay, this is representation F1. So F1 progeny of this pair of contrasting traits, it produces only one of the traits. Only one of the trait is expressed in the F1 generation. Okay, நம்ம இந்த tallness எடுத்து dwarf எடுத்தோம். இதுல ஏதோ ஒரு character மட்டும் தான் அந்த F1 generationல express ஆகுது. So he has found why it is happening. I have crossed the two different contrasting trait. Okay, a pair of contrasting trait, but only able to get one trait in the F1 progeny. So thereby he has predicted. So that means one factor is dominant over the another factor. So one factor is dominant over the another factor. Okay, one factor mask over the another factor and not allowing to express. The expressed factor is called the dominant factor. The expressed factor is called dominant factor. The factor which is not able to express is called recessive factors. Okay, so this is number dominance. So this is, we call it a dominant trait. This is a recessive trait. So dominant. Do you understand what I am saying? When we take the pure breed of a pair of contrasting traits, one pure breed on the tallness in the character on the trait, tallness always contrast on the dwarf. So when we cross, allowed for the cross pollinations, when we done the cross pollination, the F1 progeny shows only tallness. He was thinking how it is possible. So he has come to the conclusion one factor must over the another in F1 progeny. So the expressed factor in F1 progeny or the expressed trait in the F1 progeny is known as a dominant. Is known as dominant. So thereby he brought the first law, the law of dominance. When two pure plants are crossed with a pair of contrasting trait at the end of F1 generations, and only one of the trait is expressed. The expressed trait is known as dominant trait. The trait which is not able to express, and that is called a recessive trait. So if in F1 progeny, always one of the trait will be expressed, not the both. But look at this genetic makeup. This genetic makeup, one is in capital letter T, another one is in small letter t. The genetic makeup is different. It is not homozygous, it is an heterozygous hybrid. Though the genetic makeup is an variant, heterozygous, but that the phenotype, what is expressed in the F1 generation, shows one of the trait, that is in tallness. Then he thought, okay, well, let us do, then we will find out whether this continues in all the generation. So thereby he went on. He has taken this F1 progeny. So F1 progeny, he has taken F1 progeny, that is an T. Okay, T and T. So he allowed for the self-pollination. Allowed for self-pollination. self pollination occurred. This is a self pollination. Okay. When he allowed for the self pollinations, so he is able to get the dominant factor as well as the recessive factors. He thought in the F2 generations, surely he will get only the dominant factors, but to his surprise, he is able to get the recessive trait also, the recessive factor also. So thereby, so what happens here? This is, we call it a Punnett square, okay, for the calculations of the traits. Here this is considered as a male, okay, and this is we consider as a female. Okay, so what happens now? So these factors fuses with the T, so thereby it forms. Look at that F2 progeny. So F2 progeny, we can find out tallness as well as dwarf. Okay, tallness as well as dwarfness. So look at this Punnett square. So here you are able to identify the homozygous tallness. Okay, then we are able to see heterozygous tallness. Then we are able to see and dwarf, homozygous dwarf. In the first F1 generation, the dwarf, the recessive trait is not expressed. But in F2 generation, it is able to express itself. So thereby, we can get it a 3:1 ratio. So for every 3 plants, 1 recessive traits are available. So 3 dominant plants, 1 is recessive plant. So here you see this, the homozygous tallness, because the alleles are same, similar. So here you see the factors or alleles are not similar. So however not similar, they show only tallness. So this is heterozygous tallness, homozygous tallness, heterozygous tallness, and this one is an homozygous dwarfness. So what is the ratio? Phenotype ratio. What will be the ratio? Phenotype ratio. So phenotype ratio, phenotype of monohybrid, then what we call it, the F2 generations, we get it as 3:1. The tall, 3 tall plants, 1 is a dwarf plant. Okay, and another one, you see the genotype of monohybrid. So you will get it in homozygous tallness, heterozygous tallness, homozygous dwarfness. So 1:2:1 ratio. This is then monohybrids. When 2 pure plants are crossed with a pair of contrasting traits, in F1 generation only one of the trait appears, but in F2 generation there is recessive traits as well as the dominant traits also appears. Okay, this is a ratio for phenotype ratio in monohybrid is 3:1, 3 tall plants and 1 dark plants. In genotype, 1 homozygous tallness, 2 heterozygous tallness, and 1 is an homozygous dwarfness. Do you understand the ratio? Monohybrid ratio. So I have taken the characters, that is in tallness, the height of the plant, that is in one pair of contrasting traits. Do you understand? So when Mendel has done, so now we will have the clear pictures of monohybrid. So in monohybrid experiment, Mendel he has taken pure parent, that is in P. Okay, pure breed. We call it pure breed. So he has taken the character is in height. Okay, the character is height. Height in the character is pure breed. So it is having the two factors. That factors are similar. That is a homozygous allelic pair. Okay, pure breed of tallness. This is a tall plant. And pure breed dwarf plant. So what will be the factors for the trait? That's in small letter T. Okay, small letter T. At the gamete formation, only one gamete enter from the homozygous. Okay, at the gamete formations, so only one gametes enter the homozygous. From this also only one gametes enter for the cross. So this is allowed for cross pollination. Okay, now they fuse and they forms. This is an F1 generation. You understand? So he has taken in monohybrid, he has taken two pure breed of pea plants. Okay, with the only traits called, that is in height. So a pair of contrasting traits are taken. Only one pair of contrasting traits are taken for the experimental purpose. So these tall plants having the factor, the homozygous factor, capital letter T and T. Clear? The pure breed dwarf plants is having that small letter T and T. Clear? These are because factors always in a pair. So at the time of gamete formation, okay. So at the time of gamete formation, one of these enter in the gametes. That's it. So that's why I have written only T. So one of the factors enter into the gametes of male, and if you consider this as females, then that is also same. So the gametes enter, the one factor enter into the gametes. Then these gametes, the male and female gametes are fuses and forms the progeny, that is an tall plant. So in F1 generation, only one of the trait is expressed. So thereby that Mendelian has termed the predicted. So in F1 generation, whatever you take, only one of the trait will be expressed. But later on, this one is not agreed with certain conditions. Okay. So because they have taken that 4 o'clock plants, in that red and white, when it's mixed up, it gives the intermediate color, the pink colors. Okay, so this is not applicable, but however his basic idea is most probably suitable to different experiments. So that's the basic concept, law of dominance. So in law of dominance, when a pair of contrasting traits are crossed with each other, they have one progeny which is only expressing one of the traits. So that's why only one of the traits will be expressed in the F1 generation. When this F1 progeny is allowed, when F1 progeny is allowed for the self-pollination, okay, the seeds are collected from the F1 progeny, allowed for the self-pollinations. So the seeds are collected from the F1 progeny and cultivated from that plants. Again, the self-pollination take place. So F1 into F1, self-pollination. Self-pollination. Okay, self pollination button. What are the factors? These are the factors. Okay, so thereby at the time of the gamete formations, so the factors separate out, then they fuse randomly. They fuse randomly to form a traits. So here you consider this a male. Okay, so thereby this female, same thing. Now these factors are randomly reunited. So take this one capital letter T and unite with the female gamete. So thereby it forms an the same things, capital letter T and small letter t. Again you take these factors and it randomly reunites and T. Okay. So in F2 generations, we are able to get recessive trait as well as the dominant traits. Okay. So but the reappearance of recessive traits take place in the F2 generation. So thereby we can get it the 3:1 ratio of the tall plant and dwarf. So the recessive traits reappeared in the F2 generations. Do you understand? The monohybrid ratio, you should know the monohybrid ratio. Okay, phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. Got it? So what was the founding of the Mendelian experiments by using the tallness and dwarfness? Around the 747 tall plants are identified in that. Okay, got it? So we will continue in the next session. Thank you.